we have covered the basic terminology behind genetics. We've talked about what the words mean. We've talked about meiosis, which forms the underlying basis of how we can predict what's going to happen in the future, what we're going to see in future generations. Now we're going to get into the mechanisms by which we can detect that, and we're going to use some tools called Punnett squares. So for this mini lecture, we're going to discuss how to predict monohybrid crosses. One character with two traits. We'll start by using Punnett squares. Punnett squares are basically the number line of the genetics world. Just like you use number lines in kindergarten and first grade, and I don't know how far you use them. Some of you may still be using them to count the numbers going forward with addition or the numbers going backwards with subtraction. We can use Punnett squares to help us predict what's going to happen when you take the gametes from two different organisms and cross them together. So this will help us predict the crosses of two different parents, diploid parents. The way we're going to do it is I'm going to just walk you through an example. This particular example is about summer squash. So white fruit color Q is dominant over yellow fruit color little q. When I say Q, I mean capital letter Q. It's dominant. When I say little q or a little whatever, I mean a lowercase letter, it's recessive. If a squash plant is homozygous for white, is crossed with a plant homozygous for yellow, what will be the phenotypic and genotypic ratios for the F1 generation? Okay, first things first, we need to take these problems one little bit at a time, one sentence at a time. Figure out what you're given and write it down. Now, I've been doing this for a long, long time, not longer than I can remember. I can remember a time before this, but for a very long time. And I still make sure to write down all of my alleles to begin with. So I know that white fruit color is going to be big Q. So white fruit color is Q. So I write Q equals white. That's dominant, so it's a capital Q, over little q, which equals yellow. So I wrote little q equals yellow. The next step is to determine the genotypes of the parents. The genotypes of these parents, it says, if a squash plant is homozygous. Remember in the last lecture, homozygous, homo means same. So they have these same two alleles. And it's homozygous for white. Well, we wrote down that white equals big Q. So we have homozygous, two alleles that are big Q. It's crossed with a plant, so I'm just taking these one little bits at a time. That is homozygous for yellow. All right, homo means same. Our yellow allele is little q, so you have two alleles that are the same that are little q. We now have our parents. This is our P generation. Big Q, cross to little q. The next step is to determine the gametes, the parental gametes. Well, if we know our diploid gamete for our homozygous dominant is big Q, as it goes through meiosis, it's going to split and become two haploid cells. One big Q goes into one cell, the other big Q allele goes into the other cell. Remember, these are just alleles that are on a homologous pair of chromosomes. The chromosomes split and go into daughter cells. So we have one big Q allele and one big Q allele for one parent. For the other parent, we have a little q and a little q. They, as the diploid, they split into haploid cells, and you have a little q in one haploid cell and a little q in the other haploid cell. Now we start drawing pictures. Draw a box, put a cross inside of it. You need 4 boxes, all arranged like this. Then you're going to place the paternal gametes at the top of the boxes. Now, it doesn't matter if they're paternal or maternal, if from your mom or your dad. At this level, it doesn't matter. Typically, the convention is if you're dealing with plants, you put the male on top. If you're dealing with animals, you put the female on top. But that's not important for this level course. So take one of your parents, put them at the top. We have one gamete from Q, big Q, and one gamete from that same organism, big Q, at the top. Going down the side, we put the gametes from the other parents, a Q and a Q. So now we have a total of 4 gametes laid out, 2 from each parent. Each parent's gametes take one side of this square. Then you do that fertilization event. You take the sperm from the top, the egg from the side, they come together and they fuse to make big Q and little q. They do it in the next. You go to the next box. Do the same thing. Take the top one down, the side one, bring it across, big Q and little q. As you're writing these, it's kind of important. That's not important, but it's convention, and it will make your life easier to list the dominant allele first. So usually you put the dominant allele first and then the recessive allele, because the way we read in English, you read from left to right. It makes it easier for you to interpret that when you're looking at this Punnett square, you can see all of them have a dominant allele in them. Next up, you determine the ratios, the genotypic and phenotypic ratios. So the genotypic ratios, where we're looking just at the genes involved, just at the genotypes. We count the boxes that have how many are homozygous dominant, how many have big Q, and the answer is 0. Cool. Well, how many have big Q, little q? They're heterozygous. If they're heterozygous, we can count and we see 1, 2, 3, 4. So all 4 of them are dominant. You could put 100% dominant. I'm sorry, 100% heterozygous. You could put 1 to 0 heterozygous, because there's only one heterozygous at 100%. Or you could put heck, even 4 to 0. Just count them up and say 0 anything else. Typically, you're going to use the reduced number when you're putting a ratio. So instead of it being 4 to 0, it would just be 1 to 0. Instead of it being 2 to 2, say, it would be 1 to 1. So you got the heterozygous dominant. The heterozygous, and then you've got the homozygous recessive. How many are there? There are 0. Great. We got the genotypic ratios. 1 to 0, heterozygous. They all appear dominant. Or 100% heterozygous. As far as the phenotype goes, what do each of these look like? So now we just have to, for simple Mendelian dominant recessiveness, we need to look at them and say, cool, do we have a dominant allele? And if we have any dominant allele in that pair, it means that they are going to look like the dominant phenotype. Our dominant phenotype in this problem was white. So in this case, every single box has a dominant allele. So every one of these organisms is white, even though they all have a recessive allele in there. So we'll do a few more problems. We'll work through a few more of these. These are cattle. We've got hornless, big H, in cattle is dominant over horned, little h. A homozygous hornless bull is mated with a homozygous horned cow. What will be the genotype and phenotype of the first generation and the second generation? So this one isn't just asking what's going to happen in that first generation. This is asking what's going to happen when the kids mate in the F2 generation. So we start off with the problem. Again, the best way to do these problems is to take them one sentence at a time. Hornless in cattle is dominant over horned. Okay, step 1. Label your alleles. Always label your alleles. Big H is hornless, little h is horned. We got that from the first sentence. Moving on. A homozygous hornless bull is mated with a homozygous horned cow. So that tells us about our parents. Well, not our parents, about the cow's parents. I don't know, maybe you're a minotaur, whatever. So you look at the parents and it says homozygous hornless. If it's hornless, that is dominant, and homozygous means the same alleles. So it's big H, big H. And then you've got it's mated to a homozygous horned cow. If it's homozygous, it is fully recessive. It's got two of the same alleles, little h, little h. Cool, we've got our parent genotypes. Our next step is to list our parent gametes. So you take the big H and the big H, and the chromosomes that they're on split during meiosis to give you two gametes. One gamete that has a big H, one gamete that has a little h. Then your other one, your other parents, also forms gametes. A little h and a little h split to give you a little h allele and a little h allele. Great, so now we've got our gametes. We can put them into our Punnett square. For our F1 generation, we put one parent at the top. In this case, I put big H, one parent along the side, little h. And so we've got, when we fuse them together to create zygotes, diploid zygotes, the big H comes down into that first top left corner, the little h comes across, and we get a big H, little h. We do that for each box. Fuse them together. And you end up with a genotypic ratio of 1 to 0. Every single one of these is heterozygous, just like in our last problem. And our phenotypic ratio is 1 hornless to 0 horned. Can we stop there? No, because we look at the rest of the question and it says, tell us about the F2 generation. Okay, F2 generation. We start with the parents are the siblings of the F1. I know it sounds crazy. We're dealing with animals and plants. We're not talking about people. Stop snickering. We're going to take the offspring here, the siblings, in that F1 generation, and you cross those together. They become the new parents. So the P generation creates the F1. The F1 generation mates together to create the F2. So now our parent genotypes are big H, little h, cross to big H, little h. The gametes can form. You take the big H, little h, you split them, so you have a gamete with a big H and a gamete with a little h. You do the same for the other parent. It's big H, little h, so you got a gamete with a big H and a gamete with a little h. Create your Punnett square. You put one parent at the top, you put one parent down the side, and you've got big H, little h, cross to big H, little h. And now you've got some different genotypes in there. You've got a big H when they fuse together, a big H, little h, another big H, little h, and a little h, little h. So we can count up our different gametes and create our genotypic ratio. And what we find is we have one homozygous dominant, two heterozygotes, and one homozygous recessive. 1 to 2 to 1. If we're counting phenotype, on the other hand, we're counting how many of these are horned versus how many of them are hornless. Well, we know anything with a big H is hornless, so we got 1, 2, 3 hornless compared to 1 horned. So our phenotypic ratio is 3 hornless to 1 horned. Let's look at a similar problem, but we give you a little less information in it. We got in tomatoes, red fruit is dominant over yellow fruit. A plant that is homozygous for red is crossed with a plant that has yellow fruit. What will be the genotypes and phenotypes of the P, F1, and F2 generation? Okay, so in this case, we're told we're going to break it down just like we did in the last problem. I'm going to switch slides. We're going to take a look at it. We've got the problem written out. Let's take it one sentence at a time. Step 1 is always write down and define your alleles. So we got a big R allele and a little r allele. Big R is red, little r is yellow. From there, we look at our parents. One of our parents is homozygous for red. So we know for sure homozygous means same. So we got two of the same alleles, big R and big R. Our other plant is yellow. We're not told if it's homozygous or heterozygous. But because it's yellow, what is the only possible way this plant can be yellow? It's if it has two recessive alleles. So even though we're not told it's homozygous recessive, we know it is homozygous recessive just by the fact that it is phenotypically recessive. The only way it could be yellow is if there are no big R alleles. So our two parents are big R, and little r. And then we start, it looks almost exactly like the last problem we did. We've got gametes that form, the big R and the big R split to create a gamete that's big R and a gamete that's big R. The little r split to create a gamete that's little r and a gamete that's little r. We put it into our Punnett square and we see the entire F1, unsurprisingly, is heterozygous. If they're heterozygous, that means that, so it's 100% heterozygous, a 1-to-0 ratio. It also means that there's a big R in every single one of these. That means that it must be red. So it is a red, 1 red to 0 yellow plants. We move to the F2 generation where we take the F1s and cross them together. Your parents then is big R, cross to big R, little r. You create your gametes, a big R and a little r, a big R and a little r. And you end up with the same ratio we got in the last problem. It's a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. Why did I do basically the same problem twice? It could be because I forgot that I did the other problem when I made these, or pedagogically, and pretend that I planned it this way, it's to show you that you have repeating patterns in genetics. Whenever you cross a fully homozygous dominant organism to any other organism, every single offspring will look dominant. It will have that dominant allele. Whenever you cross two heterozygotes together, you will always end up with this 3 to 1 ratio with a simple Mendelian dominance cross like this, or a 1 to 2 to 1 ratio with genotype. Let's do another problem. You've probably seen something like this before. Brown eyes in humans is dominant to blue eyes. A brown-eyed man whose mother was blue-eyed marries a brown-eyed woman whose father had blue eyes. What's the probability that this couple will have a blue-eyed child? I have seen these problems. Don't even start trying to do this. Stop. Just listen to me. I'm tired of problems like this because people try to make genetics much simpler than it is. Eye color is not a simple Mendelian dominant recessive relationship. It's called a polygenic or a polygenetic trait. You cannot use simple Mendelian dominance recessiveness with human eye color. Not all genes fall into this pattern. And later on, in a few of these mini lectures forward, we're going to talk about all of these exceptions to the rule. But please note that Mendelian dominance recessive is what we use to teach the basics of genetics, because it's so easy to see, but it does not describe everything in modern genetics. You cannot do an eye problem like this for humans. Okay, that's my little mini rant there. If you've been with me, eye color in genetics and mitochondria as the powerhouse of the cell are my two big pet peeves. And I hate those. When you don't know what a parent is, so let's say you have a fully dominant looking parent, in that last problem before the eye color thing, because we're never talking about the eye color thing again, you would have a red fruit. And if you weren't told it's homozygous, you wouldn't be able to know what its genotype was just by looking at it. You wouldn't know big R versus big R, little r, or if it was big R, big R. You couldn't tell by looking. The only way you could tell what that genotype is by genetic testing, which can be expensive, or by conducting what's known as a test cross. If you crossed the unknown, the red plant that you have no idea what it is, to a known genotype, the only known genotype we have by looking at it is fully recessive, so in that case, a yellow plant. You would end up being able to see the offspring. And if any of the offspring were yellow, you would know that adult, that plant that you were working with before in the P generation, the red plant, is heterozygous because it had to have passed a yellow allele onto its offspring. because the other plant's only passing yellow alleles down. So a test cross will let you predict a parent genotype based on the offspring. People have asked before for a few more practice questions. I've added these at the end, so you can give this a little bit more practice. So there are a bunch of things going on with this mini lecture. The big thing is to look at process. Don't get bogged down in the specifics of the problem. Take it one sentence at a time. Don't try and digest the whole thing at once. Then, step 1, label your alleles so you don't get them confused. Then you identify the genotypes of the parents. Then you figure out what gametes come from that genotype. Then you combine the parents' gametes in a Punnett square to create the F1 generation, and then you interpret the Punnett square to create genotypic and phenotypic ratios. These content review questions are here to focus your studies. In the next mini lecture, we're going to start talking about how to apply this more broadly to dihybrid crosses. In this lecture, we talked about monohybrid crosses, one character with 2 traits. In the next one, we're going to talk about 2 characters with a total of 4 traits, so it gets a little bit more complicated.